Good evening, how are you guys today? I was not expecting the pizza, I just have to say thank you very much. So I'm just curious, of this group here, how many of you guys are math majors? Just raise your hand. Engineering. Biology. Okay. Chemistry. Earth science or environmental science. Undeclared. Something not in science or math. And then just say it if I've forgotten. Physics. Physics? Okay. And what am I missing? Computer science. Okay. Wow, we have a really nice, diverse group. Okay, so I'm going to talk to you a little bit about what my path was to get to where I am now in terms of my career. That includes my life experiences as well as the education that I've gotten to get to where I am. I'm going to talk mostly about research that I've done in the past, both as a master's student and in my Ph.D. program, and now as a research scientist at Ames. And then I'm going to give you guys advice, which you can ignore, or you can take it, it's up to you. And then I'm going to talk a little bit about being an intern and what that means. I've been very fortunate to have some really awesome interns, so I can tell you kind of navigating that world. And then I'll take questions from you guys. If you want to ask me questions during the time that I'm talking, just make sure to raise your hand. I get kind of on a roll. Shout out, Sherry, if I'm ignoring you. Okay, I will get to you. Okay, so what's it like to work as a biological oceanographer? Biological oceanographer. How did I ever end up in Silicon Valley as a biological oceanographer? Well, my path here has been long and very crooked. And I will talk to you about the many mistakes and learning experiences I've had to get here, talk to you about my research, the advice you can ignore or take, and then questions. So here's some basic information about me. I come from a bi-ethnic relationship. My father was from Colombia, South America, and my mother was Scottish, Irish, and a lot of other who knows what, in Illinois. My parents believed in education, and they frequently instilled in my sisters and me that they believed that we could get an education. One sister became an artist after getting her Ph.D. in cell biology, and one sister is a database or data and business architect for data management. So we kind of diverged into different directions. I'm married and I chose not to have children, but I used to be a teacher, so I consider the people I mentor and my students kind of my kids. So my sisters were born in Colombia, and I was born in Wisconsin, and we moved about every 3 to 4 years. So when I had to fill out my residency information, when I went to, San Jose State, Moss Landing Marine Labs, it said any place you've lived for more than a month. And because I had lived one place, gone back, lived one place, gone back, I had to repeat it. So there were 23 places on my list of where I had lived, of all of them, 11 states. So I've lived in a lot of different locations, but I have to say I think California is one of the best places I've ever lived. I don't speak Spanish, even though my parents were bilingual, and my mother was a Spanish teacher, and yet I don't speak Spanish. I've spent 8 months at sea or doing land-based small boat field work, and I'm extremely seasick. In fact, I often get sick driving myself to work. So there's a good chance if I'm ever arriving someplace, I'm sick. So that means that 8 months of my life I've been sick, and also collecting data and doing science. And the thing I've learned from that, 2 things. 1, get instruments that you can program. And 2, eat chocolate. It's good both ways. Okay, so as I mentioned, my path has not been a straight line. I've taken many crooked directions. I've made choices. I don't regret the choices that I've made, but I was not the type of person who went straight into college, straight into graduate school, and then straight into the research. I did a lot of different things along the way. I made mistakes, and I learned from them. And I consider myself a work in progress. And so it gives myself a little bit of a relief to know, hey, you know what, every day I'm learning. And every day I may make mistakes, but I can learn from those mistakes and I can make changes. I've also learned that I can work really hard and I may not get what I want, which is kind of a hard lesson to have. But if you work hard and people take notice, doors will open for you. They may not be the doors that you thought would be open for you, but doors will open for you. So how did I get interested in the ocean? Well, my parents thought that all 3 of the daughters were going to go on to medical school and become doctors. And so we got a lot of pressure to do science because my parents, my father being an immigrant, was very concerned with us being financially self-sufficient. And so it was, doctor, you're gonna be a doctor. Oh, I'm interested in art classes. No, you're gonna be a doctor, you're gonna be a medical doctor. And that was the path that we were supposed to take. And my older sister, who got her Ph.D., I mean, she didn't become a doctor, but not a medical doctor, in her sophomore year of college realized this is not for me. And my parents had a really big rethink of, hey, we're pressuring our kids to study something that they don't really want to study or be. So let's take a step back and let them decide what they want to be when they grow up, if they ever grow up, which is a work in progress. So what I discovered is the science bug had already taken hold. I was really fascinated with anything that was crawling or swimming. And I went on a field trip when I was in 8th grade. We lived in San Antonio, Texas. We went down to the coast and we went on a boat. This is probably not the same boat that we were on so many years ago now, but I don't know if my cursor is gonna, what? There it goes. And they put down the A-frame and they put out nets and trawlers, like doors that were running along the ground, or the benthos, the seafloor. And they brought up a squid, and they put it under the dissecting scope, and this is what I saw. And I said, oh my gosh, what is that? And it turns out that it's these structures within the squid, called chromatophores, that change in response to the behavior of the squid. And they absorb and they reflect light in different ways. And I had no idea that the seeds were being planted then to say, you know what, you're not gonna be a doctor, at least not a doctor for medicine. You're going to go into something that's a little bit more related to earth science and the environment. So I went through high school and I went to college. I went to Smith College in Central Massachusetts, the women's college, still thinking I was going to be a medical doctor. And there they had a marine science program, and I got to go on field trips and other earth science adventures, like going out and instead of going out partying on a Friday night, we went out to go look for frogs and the mating pairs of frogs and the egg sacs they were producing in these ponds. I was a science geek. I loved it though. And then I graduated from Smith and I went on to a program called Teach for America, which way back then, this was only like the second year that Teach for America existed. And I taught high school biology in South Central Arkansas, and I taught high school beginning and advanced level biology. And I had no funding, but it was a fun adventure to come up with creative ways to get field and lab experiences to the students. So it's scrappy and resourceful. After Teach for America, I got married. That's the ball and chain over here on this side. So I got married pretty young, and I moved out to the Bay Area, and I worked as a temp for a long time as an administrative assistant for a company called Silicon Graphics. This kind of doesn't really exist anymore, and we worked on an Oracle project. And I accrued a lot of debt as a student, and it was really important to me to pay off all my student loans. And so I worked really, really hard, and I was able to pay off all my student loans that I had from Smith. And I also was able to earn enough money that we could put a down payment on a house. This was so long ago that houses were actually still affordable back then. During this time, the internet boom was happening and everyone was so rowdy and crazy, but I realized this is not the world for me. I want to go back to doing marine science because that's what fills my soul with happiness. And so I applied for a program at Moss Landing Marine Labs, which is a part of the Cal State system. They offer master's degree level work. After I finished my master's degree, I went on to UC Santa Cruz. I'm a banana slug. And then after I finished my PhD, my husband and I took a year off and traveled the world in camper van. Australia, New Zealand, went to Hawaii, and went all over the western United States. Took a little sabbatical before I actually got like a real job. During the whole time that I was in graduate school, I was a volunteer at the Monterey Bay Aquarium, cleaning up fish poop and, bird poop and things like that. And then now I'm at NASA, and the reason why I'm at NASA you'll understand in just a few minutes. Why would a biological oceanographer work at NASA? You guys study space. I'm also a part of an organization now called Elevate Tutoring, and if you'd like to ask me more questions about it, it's a scholarship program, and it's a really exciting program, and one of the people who's in your program here is actually one of our tutors now. So what do I love about my career? Well, one of the things I love about my career is I get to go out in the field and take measurements and observe the Earth. And like I said, most of the time I'm feeling like I'm going to throw up most of the time. However, it's still a lot of fun to go out and be on these ships. I've spent 8 months at sea or doing field work. One of the ships that I was on for about 5 months, not all at once, was the RV Wacoma out of Oregon. The Point Sur, which is now retired, that's out of Central California. The Martin, I've gone all up and down the Central California coast along Big Sur in this boat, taking samples. And then it's fun when you get to go to the places that are in the beautiful tropics, and this is Lee Stocking Island in the Bahamas. And so everywhere you see a pin on this map are places that I've had the opportunity to go out in the field and take measurements, either scuba diving and taking measurements through that, or throwing electronic instruments over the side of the boat and bringing them back in. We like to get them back because they're expensive, but they also have the data. What is it that I like about my field of study? Well, maybe I'm a little bit distractible, maybe I have some attention deficit issues, but I love being able to study a subject where it incorporates so many other pure subjects. So what I do is I work with optical equipment, I work with understanding light properties within the water column. I have to have an understanding of the fluid dynamics of the ocean water and the movement of currents. I do a lot of work with chemistry, biology, that's my background. I actually can solder things and put instruments together and code those instruments, deploy them, test them, and bring them back in. I do a lot of coding, I do a lot of statistics and data analysis and other deterministic modeling. And some of the work that I do has implications for natural resource management and also for public health. So this inspires me because I get to touch on so many different subject areas and find where a couple of subjects come together, what's the margin there, and how can you think creatively and ask questions, science questions, that incorporates more than just one topic. And it's really exciting to be able to be in these kind of these interstices of these major subject areas. So my first major research experience was as a master's student, and I set out with 3 goals: to get my degree, to meet researchers in Monterey Bay region, and to learn tools to become a scientist. Down at Moss Landing, if you guys drive down to Monterey, it's the big smokestacks. That's about the location where it is. So the topic of my work was eelgrass response to carbon dioxide enrichment, possible impacts of climate change, and potential for remediation of coastal habitats. I never knew what a seagrass was. I had no clue. But if you ever go kayaking out onto San Francisco Bay or go in the Airborne slough and you look down and there are these green leaves either floating on the surface or coming near the top, that is a seagrass. It's a vegetative plant, similar to you have a vascular plant, grasses just outside the window here, and it's reinvaded the marine environment. They have very high light requirements and they're carbon limited in nature. So CO2 is in the atmosphere and these plants have no problem accessing it, but in the water, CO2 is in a different chemical format and these particular plants have a hard time accessing that carbon dioxide. They're vulnerable to degraded water qualities, so if there's a lot of material in the water, it makes the water less transparent and so they'll die off. They can be very productive. They're nursery grounds to important commercial fishery species, so like Dungeness crab or lobster, depending on where you are. And humans have done a really good job of destroying the habitat over the last 150 years, especially in the United States. These are the happy messages I'm trying to convey to you. So the questions I had was, does long-term exposure to elevated carbon dioxide concentration permanently enhance eelgrass productivity? Can we give them a lot of carbon dioxide and will it offset the light, limited light? And then can we use flue gas from the power plant in Moss Landing and feed the seagrasses with that flue gas? This project was funded by the Japanese government and they wanted to understand if they could do some sort of bioremediation in these environments where eelgrass had died off to try to increase the growth. Using flue gas as a means to at least stimulate them so they could handle these compromised waters. So as a master's student, I took on a lot. You don't have to read everything on here. I just am trying to show you that it's a very messy slide and there's a lot of material on it, and it's not quite showing up here. I designed and built a seawater system. As a master's student, I hired out people to put together my controllers so I could make sure that I was titrating the waters properly. And I hired another company to come over and to get a pump that they could put into the water. But everything else basically I did from about this storage tank onward. And that is, I designed it, I ordered all the parts, had them delivered, I did a lot of PVC gluing, and I think it may have caused some brain damage, but whatever. So I built the seawater system so that it could flow, have seawater flow into here. I could change the light quantities, I could change the CO2 concentration across the tanks, and then the water would flow out and go back into the ocean. So what I was trying to do is I was trying to see how do these plants respond to different CO2 and different light. So like I said, I built the seawater system. This is a few years back, a lot younger. And we went out and collected about 500 pounds of really sulfuric smelling mud. And when it gets on your hands, it takes about 3 or 4 days for your skin layer to wear off. So you smell like this sulfuric mud for like 3 or 4 days and people don't want to be near you. But anyway, this is science, so it's what we do for science. We planted all the plants into these nursery pots, and you can see here the tanks, this is not all of them here, and we let them grow. And so we let them grow for about 1.5 years, and partway through that time we started the experiment where we did the light treatment and the CO2 treatment. And what did we find? Well, eelgrass likes more CO2. You give them CO2, they grow. It responds to stress in different ways than what the scientific literature had stated. So there's a lot of research that's done on these dying seagrass beds, and they're measuring these particular characteristics, and it turns out they're measuring the wrong thing. And that was an unexpected discovery as a part of this project. And so how we measure eelgrass growth is wrong. And so they've had to totally change the way that eelgrass is measured in order to evaluate if we've destroyed the, or if it's compromised, if it's going to die. So that was kind of an exciting thing. I got a publication out of it, and that was my master's work. But I was not interested in studying seal grasses. Remember the mud? It was pretty disgusting. And so I was a lot more interested in that water column that was above the grass. What's the material in that water column? Why is there more of that material there? And what processes can be inferred from what material is there and how it changes over time? And so those are the questions I got a little bit more excited in. And there was a researcher at UC Santa Cruz named Ray Crudella who took me in as a student, and I started working on those questions. I'm going to just talk about one of the main projects from that work. So my mom says, "Why are you working at NASA?" I had no idea that NASA does what it does. I don't know if you guys know this either, but they have a science mission directorate, and they have 4 main areas that they cover: earth science, heliophysics or sun, planetary science, and astrophysics. And so when my nephews say, "Oh, tell me about the Kuiper Belt and stuff," I'm like, "I don't know astronomy, I don't know planetary science, I'm sorry." But in the earth science area, they look at atmospheric composition, weather, climate variability and change, water and energy cycle, carbon cycle and ecosystems, and earth surface and interior. And most of my work falls in the area where you see inside these 2 boxes. And NASA is really well positioned because they send things out to space, like satellites. What you see in this picture here is it's a few years old, this particular image. What this is it's the different satellites that NASA has sent out to do Earth observations. Terra is one of the satellites. It has a sensor on it called MODIS, and that is a sensor that I use a lot of data. It does Earth observations of vegetation and also just the color that's coming off of the water. You've probably, at some point in your life, even if it's just watching Men in Black, heard of the satellite sensor called the satellite called Landsat. And this is a suite of sensors that they've been sending up since the 1970s. And we have this really nice 40-year record now of Earth's surface change and what's been happening in terms of land use change. And so if you have a forest and you mow all the trees down and you put a farm out there, you're going to see that over time. You're going to see these changes. And it can give you some ideas of, hey, they've mowed down this whole forest, and we've seen a lot more die-off of eelgrass downstream. It's because the sediment is going into the river and it's flowing out into the water. And so being able to track those sorts of changes in Earth processes. One of the things that is really great is that we can take these global measurements and get an idea of how the Earth is changing over time. And one of the ways that the Earth has been changing over time is by the human use or the combustion of fossil fuels and the increase of carbon dioxide in the atmosphere as a result of the combustion of fossil fuels. Have you guys ever seen a plot like this before? No? It's called the Keeling curve. There was this guy back in the 50s who's like, we need to start measuring CO2. And everyone's like, okay. And he put this sensor in Mauna Loa in Hawaii on the Big Island. And he's been measuring, his group and subsequent groups have been measuring CO2 concentrations very high up, very high elevation. And what they're seeing is that over time, there's been an increase in CO2 concentration. And when you look at this type of record against more historical records or prehistoric records, this increase is actually quite fast. Anyone notice how there's like a zigzag effect? It's not a straight line. Do you want to posit an idea of why that might be? Seasons, right? See, you've seen this. I think you've seen this plot before. So anyway, we just exceeded 400. It varies, but now we're about permanently above 400 parts per million. When I did that study on eelgrass, my high CO2 level was actually below 400. So we've increased quite a lot just in the 10 years since I did the study. So NASA's really well equipped to look at things like global carbon dioxide. One of the things that I study is phytoplankton. And the reason why is because I'm interested to understand how phytoplankton take up carbon dioxide and where that carbon dioxide now as fixed carbon as organic form goes. Either is it exported to the sediments, is it transferred through something eating it into a different trophic level? What is the fate of that carbon? Phytoplankton contain chlorophyll as well as other pigments. These are optically active constituents that the satellite can see. You look outside, you see a tree. Your eye is seeing the reflectance of the light off of that tree and it appears green to your eye. The same idea exists for a satellite. It's looking down, it's tuned to certain wavelengths and different colors, and chlorophyll is one of the colors that it's tuned to so that we can estimate how much plant biomass there is on the planet and therefore how much carbon is being fixed out of the atmosphere. And phytoplankton are the first link in the food chain. You can see here, phytoplankton come in a lot of different colors. We see these nice green plants here, and California doesn't really have seasons, but if you were in the New England area, you'd see in the fall everything changes these beautiful colors. That's because those colors are masked during most of the year. Well, phytoplankton, if you had nice pure cultures like you have here in this image, you can have brown phytoplankton and green, light green, and you can even have red. There's a really beautiful red tide happening down in Monterey. If you go down there, you might even see a bright red surface of the ocean. So NASA has these satellites that can do Earth observation, and we measure chlorophyll over time. And what you're seeing here is a greening up of the northern and southern hemisphere. It's oscillating because of the seasons. You're seeing the greening up of these different ecosystems in the terrestrial environment and also the ocean. It's important to understand ocean phytoplankton because 50% of the air that we breathe, the oxygen in the air that we breathe, comes from the ocean phytoplankton. So if we don't have a good understanding of what phytoplankton concentrations are out there, we really don't have an understanding of what the global carbon budget is. I actually look at this sometimes when I'm having insomnia. So what's special about the phytoplankton? I've kind of talked about this. They fix inorganic carbon that goes into different directions, either sinking or different ecosystems. They release oxygen. They're the primary producers of that first link in the food chain. And they contain chlorophyll and other pigments that can be detected by remote sensing imagers, either satellites or on aircraft, or even more and more now on drones. So just a quick review, and I'm going to skip through these next two slides a little faster. If you've ever taken physics, you've probably seen the electromagnetic spectrum and the light as it hits the surface of the Earth. And this is the light as it hits the surface of the Earth, the visible light range in the 400 to 700 or 0.4 to 0.7 micron range. So the solar spectrum at the surface of the Earth. And you have these little gaps in here, which I'm not going to go into the details, but there are these transmission windows where sometimes light just doesn't make it through. So how does light interact with the sea to give us this ocean color or this signal that the satellite is seeing? The light comes down from the sun, it penetrates the surface, and here's a phytoplankton. And it can either be absorbed or it can be scattered by that phytoplankton. Detritus, it can be absorbed, color dissolved organic matter, or the water itself. It can also be scattered. That light scatters back up out of the surface. Just a small percentage of the light that actually penetrates the surface makes it back out. And then the sensor, I do mostly airborne science, so most of my sensors are on aircraft. That's why that's not a satellite here. But the remote sensing reflectance is therefore the color that we think of, the quantity that we think of as the color of the light. So scientists have taken these spectra that they measure. So we think of cameras as having like RGB or something like this. Instead of RGB, we measure 8 colors with the satellite that's currently up there. And a satellite that's about to go up in a few years is going to have about 90 colors in the visible range. And we take the quantity of that remote sensor reflectance and we just do a simple ratio of the two quantities, like in the 490 range and 551, and that gives us our ability to estimate chlorophyll from space. When I tell people this, like when I teach this in my remote sensing class, they're like, you're kidding. You just take a picture of 2 colors of light and do the ratio and that's your chlorophyll estimate. It's like, yeah, that's all it is. And we do a lot of empirical tests, like modeling and testing, but that's all it is the ratio of 2 colors of light to give us this estimate of chlorophyll in the ocean. And so I could say, hey, I have a chlorophyll concentration of X in this water parcel right here. And so that was the reason why we took satellites, put them into space to look at the ocean. We wanted to know what the chlorophyll concentration was so we could get estimates of carbon fixation to get a better understanding of what the carbon budget is. That's great. That's like 1978 technology, okay? We're in a new year and we'd like to know a little bit more. I look out and I see that there are trees, meadows, redwoods, trees, different types of trees, and urban areas here. And the satellite sensors that we have for land can easily distinguish that. But when I look at this water with a satellite sensor, all I can get is green. It's like looking at the ocean and all I see is green, maybe in different amounts, but I can't tell you different phytoplankton groups. And the reason why I want to know different phytoplankton groups is because it can drive very different ecosystems if I can get to that answer. And so we want to know more than just chlorophyll so we can better understand carbon uptake in the ocean, the transfer of that carbon through different processes, the flow of energy through different ecosystems. Some phytoplankton drive salmon, whereas some phytoplankton drive mackerel. And that matters to the fishing industry. And the presence of harmful algal species, one of my favorite topics. I just kind of like studying tornadoes and being excited about it. So how do we define these groups of phytoplankton? I'm kind of going down a very detailed path here. We could look at size. Phytoplankton can range in size from like a dust particle on this floor to the size of this building. That's the scale difference that you have in phytoplankton. And so the organism that eats the little dust particles is a very different type of organism than the one that would eat the size of this building, like Godzilla versus like a little tiny paramecium or something like that. And so when we think of, oh, it's chlorophyll out there, well, it can be very different ecosystems that we're talking about. And so most of the work that's done is trying to separate size. Size matters in the ocean. And we want to get an understanding of what those major size classes are. The biogeochemical role, I'm not going to go into a lot of this, but if you've taken biochemistry or molecular biology or microbiology, you've probably heard of nitrogen fixation. Phytoplankton can have different types of cell wall, not what we think of as these plants out here, but they can be made of glass or calcium carbonate, and also taxonomic group. And so my work is an interest in trying to separate the different taxonomic groups of phytoplankton based on what their spectrum is. And so algae contain diverse pigments and cell structures, and they give them taxonomically unique absorption and scattering characteristics. And so this organism over here was one of our bloom-forming organisms several years back. That's a dinoflagellate. It forms a really great red tide. I think it's actually the same species that's blooming right now. And then this organism that's right next to it is very similar because it has very similar pigments, but has totally different shape and size. And this one's really interesting because it produces the toxin that we know of as domoic acid. Does anyone here like Dungeness crab? No? Yeah, you didn't get to eat it last year, did you? No, it's because this guy was blooming like crazy and the toxin was bioaccumulating in the system, even down in the sediments where the Dungeness crab like to live. And so because of some interesting characteristics that were happening in the ocean with what we call the blob offshore, which was broken down, we had this like epic harmful algal bloom, and it was making all of the Dungeness crab toxic, so we couldn't eat it.
Is that related to this rising CO2?
I think it has more to do with the temperature being sustained. Does the temperature being sustained have to do with global climate change? The patterns of the wind patterns and temperature are consistent with what we would see with climate change, but I don't think we have a long enough record for me to definitively say that. But as far as like are you talking about ocean acidification? Does it have anything to do with that? I don't think anyone's actually looked to see if acidification is affecting that particular species of organism. Okay, so there's this diversity of phytoplankton colors, and based on whatever organism is, what taxonomic group it comes from, and it gives it different absorption and scattering properties, and it can result in diverse reflectance spectra. And so you can have very different spectra. People like to go whale watching. They hear I'm an oceanographer, oh, do you know that whale? It's like, no, but if you tell me it's this spectrum versus that spectrum, that's a diatom, and this is a dinoflagellate. And they're like, okay, not fine. So anyway, you can have this diversity of spectra, and it can help you understand what the different taxonomic groups. So what I did when I was a Ph.D. student is I invented a method to be able to discriminate these different phytoplankton groups. Discriminate in a good way, not in a bad way. So we're trying to look at different species of the phytoplankton groups. And what inspired that is we get these really awesome red tides down in Monterey Bay. And this picture is not enhanced at all. It was like looking at Orange Crush soda when we pulled the water up out of the, in the buckets. And what you do is you set it down, and all of the organisms, they vertically migrate because they're flagellated, they would all come right up to the surface. So you'd have this really nice layer of this really excited looking phytoplankton swimming at the surface, and you'd mix it and they'd go down. So what they do is they swim down to a sort of a division point. It's called the thermocline, where the warm water is separated from the cooler water below. They swim down, they get nutrients, and then they come back up during the daytime. And so they're really effective at that, and if there are enough nutrients in the environment, this particular type of organism can proliferate like crazy. And so I got really excited about that. Again, it's like telling people you study earthquakes. Red tides are really fascinating and really neat to understand. This guy's not toxic, but it can have such high biomass, it can suck all the oxygen out of the water. And so it can be implicated in fish kills and things like that. So we did a whole bunch of surveys and studies in Monterey. Here we are with one of these oceanographic instruments called a rosette, and it has a bunch of bottles on it that can close at different depths. We send it down on a wire and then bring it up, and it closes and captures water. And it also measures the temperature and salinity and a few other properties. Oops. And this is the organism that was implicated, and then it's hard to see in this picture, but it just looks like brown water. It was so thick. And then here's an image from the aircraft that was going over while we were taking measurements, and it's been kind of enhanced to pull out some of the features. But we tend to get this really high bloom feature in about this part of the bay. We also have a few farms with runoff in this area, and that seems to be feeding the phytoplankton bloom, because nutrients from the farms are the same type of nutrients that need the plants to grow there. They're also really good at getting phytoplankton to grow. And so what we found is that chlorophyll concentrations were really high in Monterey Bay in this year, in 2006. Usually the concentration's like 2 milligrams per meter cubed. You can see my scale goes from 0 to 150. We were finding concentrations that were like 1,000 in this area. It was like unprecedented. People said, your equipment's broken, it can't possibly be that high. We kept testing the equipment, and the equipment was fine. Using my algorithm, I was able to differentiate the different major taxonomic groups in this environment. It was dinoflagellates. We weren't surprised because we knew that that was the organism that was primarily out there. But we also found this bloom down where we were seeing this runoff from this farm that was primarily diatoms. And so we were able to identify it and to find proportions for it. And we also found that it was that toxic organism that was down there. So if I was out sampling to see, hey, should we close the Dungeness crab fishery, or the clams that are located near Autos, are they going to be safe to eat? And if I looked at the water, I'd say, oh, it's dinoflagellates, we don't need to worry because they're not the toxic ones. But if I actually looked at my algorithm and said, oh no, there are diatoms over here, let's send somebody out to actually see if there are toxins over there. And so this is a way for public health to keep people safe. And so this algorithm that I developed was one of the first to be able to separate these 2 major groups. They have very similar pigments, but their body shape and size is very different, and so we were able to discriminate between them. And the reasons for doing that is because we want to be able to understand how carbon flow goes through a system. You may have one phytoplankton that feeds one fishery, another phytoplankton that feeds another. We want to do it to detect harmful algal blooms and to validate ecosystem models. In the interest of time, I'm going to actually skip through these next 2 slides, but and then move on. So unfortunately, you guys are not going to get the video. So, okay, we'll get 1 second of the video, and I'll just talk briefly. So somebody was able to take just physical characteristics like temperature, nutrients that they expected throughout the world, and they were able to model what they think the phytoplankton will be. But there's no way to actually test to see if their model works unless you send people out at the exact same period of time that they're making these estimates for and take samples of the water. And so algorithms like the one that I invented, what we can do is we can actually use satellite imagery, and there's error associated with it, but these estimates of what these phytoplankton groups are from the remote sensing measurements, and use them to validate these mechanistic models that are based on nutrients, temperature, light, and then what they anticipate the grazers to be. If you're really interested in physics or applied math, this is the direction that you may be interested in going in, is earth science modeling and climate modeling, and making cool pictures from it. These are all physicists and engineers that are in this group. So some of the work that I've been doing since I've been at Ames is to try to refine my Ph.D, phytoplankton functional type algorithm, using data that we collected as a part of a campaign for a satellite sensor that NASA is going to launch in 2020. And it's called the hyperspectral infrared imager, and it just means that it's very high spectral resolution. So instead of 3 colors like our eye measures, or 8 colors that the sensors measure now, it has like 90 colors that it measures within that visible range. And what that means is mathematically I can do a lot more interesting work with those spectra than if I just had 3 data points in the spectrum. And so we took measurements, 3 years, 3 seasons, sick every time, out in Monterey Bay, in the Red Tide Incubator, Pajaro River mouth, and offshore, and over at Pinto Lake. After that first time, actually, they sent me to Pinto Lake. I don't know why, but we were right off the dock. And so what we had was a persistent red tide in October, but in the summertime it tended to not be quite so bad. And my algorithm seemed to work when we had enough biomass in the water, but it seemed to break down when we didn't have enough biomass in the water. So in October it worked pretty well, in April it didn't work quite so well. Another thing that we're really interested is in drinking water. So I do more than just the ocean environment, but with some other people I look at drinking water. Have you guys ever been to Lake Chabot in the East Bay? Have you heard about the dogs that have died? No? Okay. Have you heard about the thing in Lake Erie like 2 years ago where the whole city of Toledo had to drink bottled water for like 3 days? Yeah? Okay. So there's this cyanobacterium called microcystis, and there are other cyanobacteria that produce toxins, but microcystis is widespread. We have it all over the world. And what it can do is it can proliferate in freshwater systems, and it produces a toxin that is so toxic that it can kill you at very low concentrations. So if a dog runs into the water and just licks its fur, like to clean itself off, it can die if the concentration is high enough, and it doesn't really even need to be that high. So Pinto Lake down in the Watsonville area is one of the lakes where I've done a lot of this work with my former advisor. And what it is that we're trying to understand if there are ways that we can use remote sensing techniques to identify if this bacteria, cyanobacterium, is in the water, so that we can then send people out to sample to see if it's toxic. And this matters because places like Klamath or Trinity or Shasta Reservoir or other reservoirs that are drinking water reservoirs, if we're not monitoring for these toxins, they could be getting into your water system and they can kill you. And the interesting thing is that if you're exposed to a high concentration, you can die very fast. But if you're exposed to really low concentrations over a long period of time, it will kill you very slowly by producing liver tumors. And so it's a really fascinating toxin, and we're not doing a very good job of monitoring for it. Even that said, I would still drink municipal water over bottled water, because they're not sampling for it at all. So we were very interested to see if there were ways that we could use these remote sensing techniques to identify this organism in the water. So Lake Erie is an example of a place where they've had these toxic blooms. And what we were finding is that even though this is a freshwater toxin, fresh water eventually flows to the sea. And you usually think, oh yeah, if it's a freshwater organism, it's not going to survive in the marine environment. Well, the organism may not survive in the marine environment, but it still has toxins present in it. And so things are going to feed on it, and then higher trophic levels. So like maybe you'll have an urchin will feed on a biofilm that has this organism in it, and then a sea otter comes and eats that urchin. Well, what we were finding is these toxins were being vectored up through the food web, and it was causing organisms like sea otters, which are not freshwater organisms, they're out in the marine environment, were either dying or showing symptoms from having exposure to microcystin, which is the toxin that's produced by microcystis. And in terms of like how toxic is it, this isn't quite showing up so well, like it's more toxic than like a southern copperhead. It's more toxic than some of the cottonmouth. Yeah, this coastal taipon, it's going to be more toxic. And saxitoxin, which is also used as a neurotoxin, it's more toxic. But microcystin is a pretty nasty toxin. And it affects mammals. It doesn't seem to affect birds, and birds are really good vectors. They fly from lake to lake, and they're really good at moving it around. And so most of the water systems that you think of in California, little lakes, big lakes, like little ponds and agricultural settings, they probably have microcystis. They may not always be blooming, but they could foster conditions, temperature, nutrient, environment, where at some point it could bloom. So I'm not going to go into a huge amount of detail here, other than just to point out that we were seeing cycles of water temperature increase and decrease. We see increases in chlorophyll, and it's not coming out so well on this slide. And then we were seeing increases of microcystin. And the main point I'm just trying to bring to you here is that sometimes we have microcystin, the toxin in the water, sometimes it flattens out because the organism just may not be very dominant. But what we found is that always, in this lake, was preceded by another type of cyanobacterium that had very different optics than microcystis. And so we were able to develop this spectral algorithm where we were able to say, okay, this one's in the water, we know this next one's coming up. So if this one's in the water, you need to close this lake now because the other one's going to come up and we don't want anyone fishing, collecting water, swimming, letting their dogs swim in the water. And so this is just to point out on these top, microcystis is nice, bulgy looking thing, it floats at the surface. And this other organism looks kind of grass-like. And so they had very different spectral shapes that we were able to exploit in order to make these algorithms. So we got 2 papers out of the work, the one on the microcystis work and also the one on the phytoplankton functional type algorithm that I showed to you earlier. And what we learned is that my Ph.D. algorithm works okay, but you need to have a lot of biomass in the water, and that we can use these remote sensing techniques to forewarn us of these cyanobacterial blooms. So my advice. Have you ever worked with somebody or been around somebody who's like, you just ask why too much? Those are the people I try to avoid as much as possible because why is like the most important question you can ask, and it's a fun question. And yeah, you might get dirty and messy as you try to answer it, but like you get to learn, hey, you know, that's why. So try to ask why. When you find yourself not asking why, do something, I don't know, go for a run or something, go out and do something interesting, get yourself away from it to be able to go back and ask yourself why. Cultivate a growth mindset. Are they familiar? Do they use this terminology here? Okay. Cultivate a growth mindset. I grew up in a fixed mindset household, and I am constantly trying to be a better growth mindset person, that through hard work and effort, I can learn things. Take risks and be prepared to fail. The algorithm that I showed to you guys, that was the 4th algorithm that I came up with. I came up with 3 failed algorithms before that. It took 2 years of my PhD program to finally get to an algorithm that worked. And at first, because I had a fixed mindset, I really struggled and I just didn't want to say this one failed. So I held on to it even though it just didn't work. So be prepared to fail, and fail often, and use it as an opportunity to learn. And this last one is one thing I just didn't do as an undergraduate, is get tutoring if you need it. And that's what's so awesome about your program, is you have these tutoring opportunities, either to give it or to get it. I struggled my first 2 years of college quite significantly because I just wouldn't ask for help. There are only 2 pages of advice, I promise. Build a strong foundation in science and math. I find that at least people who are working on environmental science will say, oh, environmental science is cool and easy. And it's like, no, you need to really get a good foundation in biology, physics, and chemistry before you get into, and math, before you do environmental science. Seek paid research opportunities. Note paid. If you're in STEM, you should not be working for free for an internship. I did not do internships when I was an undergraduate, and I regret it. There were not as many funded opportunities way back then, but I also just didn't seek them out. NASA has a huge number of internship opportunities. OSSI is, and I'll send you the links. There's a program called Develop, if you're interested in that. There's a program called Student Airborne Research Program at NASA for rising seniors, so after you transfer, you'd be eligible for it. And you get to go out and fly on one of the NASA aircraft over the Central Valley and over the coast collecting data, and then you spend the rest of the summer analyzing the data. It's okay to start over. I've had like at least 3 starts. Learn and continue to refine your skills. I highly recommend that if you don't already know how to code, that you learn how to code. Learn at least even just the basics of statistical analysis. Learn public speaking, and get comfortable with writing. And I find that sometimes just writing email to people who are like friends or family helps me to come up with ways to describe things at multiple different levels so that you can convey your message to more than one type of audience. And then the last one is don't always follow the advice. It's okay to be skeptical. So I had an intern this summer who was awesome. I loved her. I was so sad to say goodbye to her at the end of the summer. She came into work every day, earlier than I did, worked almost as late as I did, and she was an independent thinker. She thought about the questions. She came up with new ideas. She challenged me. We found a mistake in her work that I had actually suggested that she do, and she said, I don't think this is working. I think that it's actually something else. And so she had the courage to speak honestly with me. And that's because she she had this willingness to like speak truth to power, not that I'm powerful, but she had this courage to speak up and say, I don't think you're right. And she was right. And it was really impressive to see that growth. And so to treat it like a job, even though maybe some of the other interns might be treating it like it's a frat party, and come in every day to work, ask more questions, be willing to read, dress professionally. I think that's about most of it. When you reach out to people, it depends on the type of internship you're getting. Don't be afraid to cold email people if you're interested in doing an internship. You have nothing to lose. And I tend to be pretty shy, but cold email people and say, hey, you know, there's this thing that you do I'm really interested in. And if you don't want to go into research, that's fine. Try to do a research opportunity, like a research for undergraduates. It doesn't mean you have to do research for the rest of your life, but it'll show a future employer that you have the initiative to look for something like this and the ability to complete a task. And you have a portfolio item that you can then take to your job interview and say, oh yeah, I coded up this whole thing and I was able to get this whatever to work. I know, not specific, but. All right, so those are my pieces of advice, and I'd be happy to answer your questions.
For about 6 minutes or so, any critical questions?
Why do you call the birds vectors? Like, is it because they what they have to?
So, I'm a birder. When I go birding, I think of them as birds. But I'm describing them in a process sense here, that they are a means for the cells to get from one lake to the next. If you had a boat and you didn't dump the ballast water, you are also a vector. Not you, your boat.
Okay.
What program do you use to code besides Python?
I actually am trying to learn Python now. I use MATLAB.
Okay.
But when I finished being a student, MATLAB goes up significantly in price. And so I'm learning Python now. Some of the coding I do is in Python. Some I do in IDL, which isn't that commonly used unless you do image processing for astronomy. And then there are other software packages that I use as well that are specific to the type of work. But MATLAB is mostly what I code in, but I would like to move over to Python. I don't do a lot of really heavy-duty biostatistics for my work, but if you're interested in going to bioengineering or into biology or bioinformatics, then the coding language R is also open source like Python is, and it has a wonderful user group that has a lot of the statistical tests already built.
Oh.
Sure. I'm happy to. There are also some links here that might be of interest to you, and I can also give these links to you as well.
Okay, so we want to thank you for coming tonight.